It's thinking about the recording. Still waiting, sorry. And so now we're recording chapter 13, spinal cord and spinal nerves. Spinal cord and spinal nerves will be this first upper track, and it's kind of good with the nervous system because if you separate off an anatomical part of it, you can actually understand something about how that works. So we're going to start right down here with the spinal cord and spinal nerves. And we're going to focus on this afferent tract sensory receptors through spinal nerves, the peripheral nerves attached to specific spinal nerves, which is their pathway into the central nervous system. And we're also going to look at processing that occurs right in the spinal cord, what we call a reflex. Under normal circumstances, you put your hand down on a hot skillet, it jerks back very rapidly and on its own. That's a reflex. And the processing is occurring right here. But we're also going to then send a message back out to a response cell. There are effectors, are three types: muscles, glands, and fat. Muscles of the three types shown here: skeletal, smooth, and cardiac. But these are the only three things that can respond when a nerve impulse goes back out. Now, after that, we're going to look at sensory receptors to what we call cranial centers. It's going to come in through, by the way, the spinal cord and up to the brain, where it's going to be processed in reflex centers in the brain, a cranial reflex, and then send the same motor outputs out to cranial nerve. So this is how we're going to start. And we're going to start by revisiting what we know about the skeleton. The axis of the vertebrate body is formed by this vertebral column from the C1 through the S5 and coccyx. The arrangement of the spinal nerves and their numbering is reflective of the organization of the vertebra. We have 7 cervical, 12 thoracic, 5 lumbar, 1 sacrum, although we're going to have 5 sacral pairs. And then down here, a coccyx, a coccygeal nerve, the final one that comes out of this enclosure. Now, this is our flexible structural axis, very strong, supports our weight, but maintains our flexibility. We begin numbering the spinal nerves, those that are outside the skull, by the vertebra that is adjacent in the cervical region, the space here between the occipital lobe and the C1 vertebra. There is a space, and the left and right nerve that connects to the spinal cord through that space is called the C1 nerve. You'll notice that the C1 nerve is above the C1 vertebra. So let's keep that up all the way down here through C7. So the C7 vertebra is below the C7 nerve. Now we're going to mix it up a little. They've decided that the spinal nerves below the C7 are the C8 pair. So the C8 is below the C7, and that organization occurs from here to the bottom. So the T1 vertebra is above the departure of the T1 spinal nerve. So if you go down here to the middle, here's the L2, right above it, this space where the pointer is where you would find the L2 vertebra. That continues all the way down to the L5 below and the sacrum. Now, we said before the sacrum was a fused bone from originally 5 vertebral segments. But one fused bone looks pretty solid to me. but those lateral holes, left and right of the midline, provide for the departure of 1, 2, 3, 4, 5 sacral nerves. The cross-section of the spinal column is shown here, with the color indicating the difference between the central gray matter and the surrounding white matter. Gray matter means naked membranes. White matter means Schwann cells, myelinated axons, and fast transmission lines. This always reminds me first of a butterfly. Surrounds the central canal on the midline and is symmetrically arranged around the midline, this anterior fissure and the posterior sulcus, dividing it into several characteristic folds. Now, one of the things that occurs with spinal nerves, notice that the spinal nerve out here, which is a single strand that we see, we call this the C3 nerve, showing right here. The C3 junction will split inside the spinal cord to a dorsal root, often with a ganglion here, and a ventral root. I'm going to jump the gun here and tell you that anatomical difference reflects a functional difference. The incoming signals of the afferent system, the sensory signals, are connected into the gray matter of the dorsal root ganglion, dorsal root connect to this dorsal gray horn. That's where the sensory messages are coming in. In this gray matter on the right side, you will see synapses that distribute those sensory inputs and send them on to all levels of the body. When a motor response is generated, if it's going back out through that same junction, it's going to go out through the ventral root. So dorsal, sensory in, ventral, motor out. You see the different sizes of the different levels. Sacrum gets pretty small. In general, if there are more nerve connections, the cross section of the spinal cord increases in size. You can see a blow up of that here. The dorsal and ventral root, and again, right and left side, always reflect sensory in, dorsal, ventral, motor out. Gray matter and white matter is always recognizable by its difference in color, but that just begins to summarize the diversity and the variety in the spinal cord. I like this diagram very much. It actually shows you how the spinal cord, its gray matter and white matter, interacts with these roots. So here's the spinal nerve. Since this is the back, this is the right side now, and this is the left. You can see here the dorsal root and dorsal ganglion coming in, and once it gets inside these wrappings, it splits, it branches, so that it's actually connecting to the dorsal, the dorsal gray root of the spinal cord up and down all across the expanse of the vertebra that encloses it. So everywhere between up here and down here somewhere, we're going to get connections from this dorsal root. Processing goes on in this gray matter and gray matter in the brain, and sends motor commands as necessary back out through this ventral root and out the right side. Notice that it's reflected by a similar anatomy on the left side, the mirror image. The spinal cord is enclosed and protected by 3 membrane layers called the meninges. The meninges separate the central nervous system, brain and spinal cord, and create a physically protected and isolated, as well as a chemically regulated internal space in the central nervous system. We do not have blood as the main fluid. We have cerebrospinal fluid. Although blood flow brings oxygen and glucose into the brain, The neuroglia regulate its passage to the cerebrospinal fluid side where it can interact with the cells. Right here on the outside of the perimeter of the white matter of the spinal cord is a closely adhering membrane called the pia mater. It's protective, and as you can see, it provides structural support for the blood vessels that need to penetrate this tissue. The arachnoid mater is actually a series of spaces and kind of crosshatches that provides a space between the pia mater and the outer covering, the dura mater. Now, the way this is cut and reflexed, it looks kind of curly. It looks kind of like it's a stretchy, strong membrane, and that's exactly what it is. Dura mater is very tough, very flexible, but also almost impenetrable. And it is what provides the main protection and covering and the main isolation of the central nervous system from surrounding tissues. You can see in the way the spinal cord in its wrappings down below nestle into this vertebral foramen, and also how these lateral holes that basically exist between one vertebra and another provide the openings for the spinal nerve to connect the periphery to the internal central nervous system elements. Take a cross section, look down from the top. Here's that vertebral body. Here is that posterior spinal process, spinous process. And you see the butterfly-shaped gray matter and the surrounding white matter of the spinal cord. Around that you can see black. And the black is a space filled with cerebrospinal fluid. And the pia mater is right where the pointer is right there. The arachnoid mater is the black space in here, maintaining that space open for fluid content. I'll get back to that in a second. And the dura mater is this blue outer covering that is right next to the fat packing that separates the dura mater from the bony, hard surface of the vertebra. Now, this black, as I said, is filled with cerebrospinal fluid. It's not just nourishing. It's not just a diffusion medium for oxygen and glucose. It is, in fact, a hydraulic shock absorber. So this gives the spinal cord the ability to move somewhat around in this space, but when it tries to shift in this space, the fluid resists it and cushions it all up and down its length. Shown also here are these white strands and this posterior strands, the denticulate ligaments that basically connect to the bony structure and suspend, based on connections to the pia mater, suspend the spinal cord and hold it within its cavity. Here is what it really looks like when you dissect the anterior side. This column in the center is the spinal cord. See its limits. And we have the anterior median fissure, that prominent space between the right and left side is clearly visible up here where they have peeled the pia mater back. Can you see this pia mater surrounds and clings to the outside of the spinal cord? You can see it also suspends blood vessels. See that blood vessel running up through there? See, it's closer to the outside surface here, delivering blood. Blood does have to flow through the brain and spinal cord. It's just those blood vessels are going to be covered with astrocytes. They're going to be covered with those Meninges cells so that a neuroglial cell regulates all the exchange with the blood flow. Now, other things you can see here, as you pull this apart, this outer strong covering, you see the back of it here. And here is the dura mater. It's been cut and reflexed back. And as you pulled it, this arachnoid mater kind of freely separated and pulled, basically pulled apart as you pulled the Dura Mater back. Also visible, you can see here, a ventral root of one of the spinal nerves. This is the motor out. So from connections between here and here, motor signals are going out through these ventral elements to the ventral root of the spinal cord, in this case on the left side, I don't know the number, and basically carrying messages out to the periphery. The concentrated fibrous white tissue, you see how it's very solid between these connection points, and it seems to form a kind of a fan shape right here, is the denticulate ligament. Here and here, kind of identifying the limits of coverage of one vertebra, because between here and here, nothing gets out. However, here is the intervertebral foramen, and here are the connection points. These denticulate ligaments go from the pia mater, where they're connected to strong fibers, out through the Dura Mater and connect to the bony structure of the vertebra that surround them. So physically and chemically, we have a lot of very interesting structure here. Now, it's very difficult. I can see here, you see this strand right up here that seems to be running around out of sight. This is the lower portion of the dorsal root. You can see another dorsal root here, labeled ducking under this denticulate ligament and heading for its posterior attachment, the posterior gray horn of the spinal cord. Now, good news. With a spinal column that is so carefully wrapped, it's devilish hard to get infection or toxin into the central nervous system, including the spinal cord. Bad news, if it does get in there, there's some kind of pathogen or toxin in there causing an infection of the three membranes, the meninges. We call it meningitis. It's very difficult to treat. When you're looking for an effect inside the spinal membranes, you do what's called a spinal tap. Now, raising the arm in this way basically causes the vertebra to separate a little more, and this needle has to go in between the vertebra, punch through the Dura Mater into that space, the arachnoid mater, where the fluid is drawn out. That fluid is cerebrospinal fluid, and it would be sent to a pathologist, where the pathologist would look for physical signals of infection or degradation, fragments, dead cells, pathogenic materials like bacteria or viruses. That's the reason for a spinal tap. It is a combination of science and art. Everyone who becomes a doctor is trained to it. You'll notice in any hospital, they recognize the few doctors who are actually very good at it, and they're often called down for emergency treatments to do the spinal taps, regardless of their floor of assignment. So here we are back for the cross-section of the spinal cord, the gray matter and white matter, and we're going to break down the sensory and the motor signals into regions of gray matter. That's done over here on the right. Over here on the left, you'll notice the dorsal root ganglion to the posterior gray horn connection. Here is the lateral gray horn and the anterior gray horn. The white column outside is called the lateral white column. The anterior white column is in front, the posterior white column in back. Over on the left of this, we've figured out the anatomical terms. On the right, we're going to color code the rough regions for these motor commands. So we have from the sensory signals, these first gray matter nuclei, more toward the periphery, are receiving somatic signals from the skeletal muscles. The visceral signals from our organs and smooth muscles and so forth are coming in this kind of, I don't know, bluish gray region. As we cross the midline that's kind of established this, not midline, but this midway line posterior to anterior, the one that would go right through the central canal. We get to this blue region, which is sending out commands to the visceral motor nuclei. Now, those are the glands, the fat, and the muscles that are in our organs, the heart, and the smooth muscle of the other organ walls. And then here, this big pink region would be connecting through the anterior gray horn to the ventral root and sending messages to our somatic motor nuclei. Remember from our original breakdown, that was the skeletal muscles. So we do see a rough organization of the gray matter for the in sensory and out motor transmissions. When you do a cut, you definitely can see the difference, even in a fixed and stained section like this one, between the central gray matter, crossing here at the central canal. This gray matter is kind of more obviously butterfly shaped. We have posterior, lateral, and anterior white regions surrounding it on each side. A posterior gray horn and anterior gray horn are really clear. But when you do this kind of section, it has to be really thin. So when you cut it, all of these delicate connections, like here and here, are often ripped apart. And that's actually what's happening. This is the dorsal root, but up here it gets fine enough that the blood basically just pulled it apart and fragmented it. The remnants that you see as kind of rough, broken lines are the remnants of the more delicate part of the meninges. Here are the remains of the ventral root, basically coalescing here. And as it becomes more solid, it's a little more solid on this side, you can see the split between the dorsal and ventral root of that spinal cord. So the histology of a nerve bundle is going to use the same prefix terminology that muscles use. You remember muscles were tubes within tubes within tubes, so are nerves. So here is a nerve drawn in its entirety and surrounded by a membrane called an epineurium. Within it, though, you see bundles called, again, fascicles. And the fascicle, shown here extended, is surrounded by a perineurium around one fascicle, and the fascicles are full of smaller tubes. These are the myelinated axons, and this is one individual axon in the Schwann cells. It is surrounded by an endoneurium. Now, you'll see it's parallel to the tube within tube within tube organization in the muscles, except we have replaced myo with neuro in order to understand it's the nerve system. Now, notice that same kind of bundle organization, a tube within a tube within the largest tube is larger than the frame here. You can see the white matter appearance of the myelinated axon sticking out. You can also see the penetration of the blood vessels, with the larger vessels being between the fascicles, distributing the main blood supply, and then penetrating to smaller vesicles that are shown inside the fascicles and providing blood to the capillary beds that will do the actual oxygen and nutrient supply, glucose, to these individual cells. Now, nerve cells are very different in that they often are very long. An individual neuron might be 30 inches long. So these are running side by side, are organized into bundles, as shown here. Now, these next drawings are going to kind of amplify try to extend this bundling and this dorsal and ventral root idea to messages from around the body. And obviously, all four drawings occur right here in this space. But for clarity, we're going to separate sensory, showing this combination of somatic sensations in red and visceral sensations in purple. You'll notice the somatic sensations come mainly from the lateral and posterior elements, so that the exteroreceptors of the body wall and of the limbs kind of come this way. The purple interoceptors, we do have interoceptors all over our body, and so we do find them posterior, lateral, and especially here anterior. This sympathetic nerve carries sensory information from the visceral organs. Notice that they do show the presence of those kind of little knots or synapses where we are dividing sensory signals and maybe sending them over many nerves. But here is the somatic nerve sensory reception, and here is the visceral. Now, if we go on, to this larger diagram, I should have gone on before. We notice that there is a kind of a structure from the back, side, and front that is collecting at each one of these vertebral junctions. So the spinal nerves are going to be collecting information in a band that runs around the body. And I always find it more appropriate to start my thinking about it with my mind back where the vertebrae are. The spinal nerve basically would send motor nerves out to that specific region and would gather sensory input from that region and send it back. Now, I'll show you these regions in just a moment. When we think about motor commands, they are going to be coming from the anterior grain horn, visceral and somatic, out through the ventral root and the spinal nerve, and often to a nearby ganglion. This is showing a sympathetic ganglion where synapses are dividing up those messages so that the message can be sent to every effector that needs to be activated. Now, this is kind of an interneuron idea, and the rerouting. So visceral motor nuclei basically go through the sympathetic ganglion and straight down to the nerves. The somatic motor neurons are going to be distributed through the posterior and lateral elements of the nerve to the skeletal muscle of the body wall and limbs. This black connection is an interneuron that actually originates receiving its visceral signals from the from the synapse here in the ganglion and distributing it literally to all parts of the body. When we talk about sympathetic nerve stimulation, we call it sometimes fight or flight. You should think of it as absolutely maximum activation of muscle, of lung, of heart, of senses like vision. And so that means once that stimulation occurs, we're going to basically use these interneurons to send that message to every one of those elements that support active physical activity. Oh, I forgot to do the blow up again. But notice that there is a suborganization of the anatomy, and it includes not just direct transmission of these images or these nerve impulses out to the effectors, but an actual another level of organization of distribution of that nerve signal so that multiple elements of the skeletal system, multiple elements of the muscle system are activated all at once. And at the same time, you can send an inhibitory message to those areas that you don't need. So if you're running for your life or fighting for your life, you'd like to divert the blood that normally occupies the wall of the digestive tract, the reproductive tract, the urinary system, divert that over to the muscles. And that's a part of this complex networking. Now, once you look at the individual spinal nerves and follow the connection to their sensory and motor elements, you get a pattern of alternating bands of innervation. We call these bands dermatomes when we draw them adjacent to one another and show their borders on the skin. So on here on the anterior side, you will notice a somewhat irregular pattern on the appendages, but a very regular kind of segmented pattern from T1 to T12 for the main part of the trunk. Now, back here where you have the spinal nerve connections, you can see cervical nerves are involved in the head, the neck, the shoulder, but cervical nerves really do have a major impact on the shoulders, arms, hands, and wrists, all the way down here. Now, you do have some thoracic involvement. You see the T1 and T2 nerve do kind of impinge on the arms, but generally, the thoracic dermatomes are following the ribs. So up here, here's the T2 region. Notice how it ducks under. See it descend? It ducks under the right armpit on this side. Here's the right armpit on this side. Comes around and kind of follows the nerve to its connection to the sternum. But look at the difference. Here's the T2 dermatome departure, and here's the T2 dermatome connection to the sternum. And that's basically a reflection that the ribs slant downward. At rest, they basically sit at an angle. We only raise them for active breathing. So these dermatomes are often referenced. Their shape is affected by presence of skeletal elements that can support them, like the ribs. You'll notice the clavicle and the shoulder blade are all skeletal elements. But they're also affected by the fact we're bipeds, standing up on two legs, and the weight of gravity is pushing toward the floor in this figure and basically stressing our skin. So we're going to find that the uppermost parts of the shoulder have the thickest skin, where the weight of the skin pulling down is heavy, the skin will get thicker. Where there's almost no pressure on the skin will get thinner. Now on a quadruped, like a cow, that means the thickest hide, that part that makes the leather for heavy straps like belts, or even heavier straps for things like harnesses for horse and buggy connections, are right along the top of the backbone. As the skin curves around to the belly, the skin gets thinner and thinner. Now, this is a very interesting picture showing a disease called shingles. These red rashy areas are areas that are irritated by an activation of the motor nerve. The inflammation response has caused a flooding with blood, which causes the redness, causes the stretch receptors to become hypersensitive, so they itch. Often they will form, in advanced cases, you can kind of see the nodules. There'll be a little knot here and a little knot here, and sometimes will form pustules. This is following the path of one dermatome and the path of a particular one or two dermatomes, reflecting the spinal nerves that are involved. Now, shingles comes from an activation of the chickenpox virus. Most of us have had chickenpox or vaccination, and when we get well, the virus remains inactive in certain locations in the body, especially at the intervertebral junctions where the spinal nerves are connected from periphery to central nervous system. So if the chickenpox virus flares up, the infection in that intervertebral foramen irritates the motor nerve, and it calls for an inflammation reaction, even though there's nothing wrong right here. Calling for a nerve signal produces these patterns of rash, itching, and unpleasantness. Activating the chickenpox virus is sometimes dependent on an event like a traffic accident, and part of the recovery would be the development of something like shingles. Another factor that figures into this is age. That shingles develops into active and irritating infections in older people. So we have overall a pattern of vertebral division of the spine that dictates the arrangement of spinal nerves up and down. The only real kind of departures from the formula are the C8 cervical nerve, which moves the numbering system from above the vertebra to below it. And down here, the 5 sacral nerves that come in pairs out of one single mature bone. The cervical and thoracic elements that serve the arm form a bundle of fibers. Now, when you have to take it across a very mobile region like the shoulder, you're going to tuck it into the very best place in terms of keeping it away from that bony joint, running it across a space where it can stretch and move, but not get pinched. Here then we have the division into median, ulnar, and radial nerves as they depart down the arm, and you'll find that many nerves in sections of the body are named for their skeletal components. This is the so-called Brachial Plexus or arm plexus. This is the cervical plexus that divides to serve the head, neck, and part of the shoulder. Down here we have the lumbar plexus and the sacral plexus, both of which are involved in the pelvic region and also into the management of our lower appendages, the legs. I'm not going to go through these plexus arrangements in great detail, but I thought it would be useful to emphasize one thing from this drawing, and that is that this is a branching tube network. Branching tube networks are sort of like the branches on a tree. You can see them in the winter. They stick out there and occupy a great volume of space. And the reason that branches do that is to fill space and provide different places where leaves can grow and remain in the light. They fill space. And so you see the departure here of the C1, 2, 3, 4, 5 cervical nerves. Now, I say departure with kind of my tongue in my cheek. Every spinal nerve that we name, so the pointer is right below the C3 nerve. The C3 nerve is the one on the right side that's above the pointer, and it contains both sensory elements that are shooting sensory messages about that dermatome into the central nervous system, both spinal cord and brain. Information processing goes on. If a response is needed, they're basically carrying motor messages back out. So these are definitely two-way streets. But one of the things that's really obvious, just from looking at this first example, is how often these nerves loop around and connect to one another. So here is the spinal nerve C2 coming out. And you'll notice this major element that loops around and connects right to C3, which loops around and connects to right to C4. So the thing I want to mention is that, like the C3 nerve does not have exclusive use of its dermatome, there's a lot of overlap, a lot of converging of messages for synapses to distribute them to new places, a lot of diverging of messages in the normal activity of our spinal cord. So you can see, I mean, every one of these drawings is kind of a study in itself. The clavicle, you can kind of see how far forward it moves to place the sternum. You can see the presence of these muscles that manage the hyoid bone, its connection up here to the chin. This is a great tissue area, but basically the bony connections are pretty widely spread. Also in this drawing, something that I just want to mention, if a pair of nerves comes off of the brainstem inside the skull, we call it a cranial nerve, and we indicate it with a capital N and a Roman numeral. There are 12 pairs, and you can see that the cranial nerves here, 11 and 12. The accessory nerve, which doesn't tell you much, but the hypoglossal means below the tongue. Here is the hypoglossal nerve, basically providing innervation to the bone, basically the sensory apparatus right under the mandible, under the tongue, and even down here, perhaps to control the thyroid muscle. Now, these cranial nerves usually are going to stick to the head and neck region. And the cranial nerves have a big influence on our special senses. Sight, smell, taste, and hearing all involve one or more cranial nerve. But even though it's within the skull, the cranial nerves are considered part of the peripheral nervous system. So I want to underline this definition because it often confuses people. Central nervous system is the brain, the spinal cord. The brain includes the brain stem, but the nerves that come off of the brain stem are the cranial nerves, and those are classified as peripheral nerves. So we don't want to, I don't want to make much of the detail of how these branching systems depart. Here's the brachial plexus. Largely, you'll notice cervical nerve crossing this opening between the clavicle and the scapula and crossing here through the axial region. Now, this is not a very good drawing because the scapula is way, way back behind the ribs. The ribs are curving way in here. This is a big space, if you'll recall. The number one rib shown here is forming a small circle and a lot of space. So the brachial plexus comes across here in three main runs: the musculocutaneous, the median, the ulnar, and the radial nerve, 4, and nerves that then cross the elbow at strategic places to keep them out of the joint. And we basically continue the naming of the radial nerve, ulnar nerve, and medial nerve across the antebrachial region before it splits into its palmar elements. Now, this basic anatomy is retained for the sensory side and the motor side, although I do want to kind of emphasize that they do retain a kind of a rough anatomical arrangement. So this color pattern shown on the hand for the brachial plexus are those areas shown in yellow are innervated by the median nerve. The ulnar nerve takes a rather long patch, including all the 5-digit and half of the medial side of the number 4-digit. And the radial nerve has just a little bit at the base of the thumb on the anterior side, but a great big part of the 1 through 4.5 and the back of the hand. So this kind of mapping is somewhat useful. Also, I want to re-emphasize this pattern of joining nerves that join together when you look into the body or when they split apart. That shows the way that nerve messages are passed from one element to another. So this is showing the brachial plexus, and the color code indicates the division and the unification of different departures of these spinal nerves. So these purple are called the roots. Now the ventral rami, shown in purple, converging here. Look at that. Those 5 elements converge into 3 trunks and then split into orange divisions in a somewhat irregular way. The blue are called cords and the yellow are called peripheral nerves. So I want you to just, in a general way, just recognize that there's a lot of convergence and divergence in these elements. So let's say I come out of C5. If I follow this pathway, I can end up in the radial nerve. If I follow this pathway, I can end up over here in the musculocutaneous or the radial nerve. Do you see that kind of branching and unification pattern? Now down here in the lumbar and sacral plexus, we have a repetition of this same kind of branching, the same kind of branching out, filling the space, collecting information from sensory receptors. You are all well aware that that sensor for heat is a very popular item. All over the body, we can sense a warm item any place on our skin surface. And collecting that information and sending it in converging nerve networks, and motor responses then are a little different. You know, you don't always send the motor response back to the location of the sensory stimulus. So finger on a hot skillet, you're going to send that message here to the biceps brachii and flex the elbow. You don't have to send a message back to where the initial burn took place. So we are seeing here some of the major nerves of the body. I reiterate that the sacrum is a 5 vertebra fusion in our body, but they have retained these lateral openings that allow the S1, 2, 3, and 4 sacral nerves, S5, to depart just as if they were still separate vertebrae. So even though the bone is fused for structural support of a biped, the pairing and the connection of the spinal nerves remains along that historical segmental organization of those vertebrae. You can see the major nerves coming down. The sciatic nerve is one that runs out of the pelvic region and all the way down the thigh. You can see the location of these lower nerves, the sural nerve, saphenous nerve, and fibular nerve, using, again, our good regional anatomical terms, show you the mapping over the foot and ankle region for sensation and motor control. Now, that's a kind of a very quick overview, and in some way, if you're a reductionist, in other words, you like to go down and get to the cellular level and get to the chemical level and say, no, show me how, when I scratch my head, show me what those nerves look like. You like to break it down to that level, maybe a little unsatisfying. So we're left with the idea of the shape of a neuron, which is diagrammed in various ways here. Those little points represent places where branching dendrites extend from the bodies, and those gaps represent synapses. And this shows a couple of ways that we envision these neurons organizing themselves. Because we know that one sensory signal doesn't just go to one place. We need that sensation to go to multiple places. So the first idea here, a single neuron and 2 telodendrians splitting to form synapses with 2 postsynaptic cells. If in fact you stimulate and reach an action potential, you've just taken that nerve impulse and split it in 2. So this is called divergence. So for example, when we send a message to the reflex center in the spinal cord, we're going to send this on to a motor processing region to initiate the action of withdrawing the hand. But we're going to split that signal and send this up through this neural network, up through the white matter that's in the spinal column to the brain, which is going to, when it reaches the appropriate place, is going to produce a pain sensation. And pain sensation is very useful. It tells you where to look right now. How much damage is there? Once pain notifies you, do you notice it kind of goes down? Pain subsides. It still is there, but it's not going to build to the place it incapacitates you. That would be counter to the kind of addressing the medical condition or the injury. Now, conversely, here's convergence, where we have these 4 blue cell bodies. Maybe these are hooked to heat receptors across the finger of one hand. So you may encounter a heat reaction over here that doesn't really reach these other 3 cells. But once you stimulate that action potential, it can go to a central transmission point and write up this column, because it is specific to a location. But the transmission out of the finger and out of the hand is one that can be shared. This is called convergence. Serial processing is just a straight line, easy to understand. Parallel processing occurs when you have a very, very dense array of neurons packed very close together. Neurons, or what are called neuronal pools, that receive constant flow of information and need to process that constant flow of information every second. So when you think about it, our sense of sight is continuous. It's real time, real space. Because of the speed of light and the speed of transmission, I'm actually interpreting a world where there's just a very tiny delay between my understanding and perception of the movement and the movement itself. So there we have to pack these divergence neurons together very closely, and we'll basically line these transmission neurons up. You think about our image of the world, sight doesn't seem to have a size. Our eyes determine our perception of the world, but that's all being processed by light falling on a sensory area that's about this big around, about as big around as a nickel to a quarter. So the detail of the light, its density and the wavelengths and how they fall into pattern requires this kind of parallel processing. Finally, reverberation is a real interesting one. Here's that first neuron stimulating the middle neuron so that it can pass the message on down the line to pink. But the purple curves back and delivers another stimulation to blue. Now that creates a kind of a cycle here where blue keeps getting stimulated. This is what we imagine for positive feedback. Positive feedback, you remember, isn't like most of the homeostatic regulation in your body. Homeostasis, typically, whatever the stimulus is, for instance, temperature in the body is going up, negative regulation would change body systems to bring it back down, and vice versa. But positive stimulation, positive feedback, is one where the stimulation builds to a conclusion. Clotting of blood, vomiting after nausea, the delivery of a baby after the reverberating and recurring contractions that produce the physical push to push it out of the womb. Now, reflexes are really interesting to me. We're actually wired to produce some responses quicker than we can recognize they are happening. I love the burn response or the tack response because actually it happens so quickly, is that my hand will jerk back from something and I feel my hand move and I've started to go, what's going on? And looking over, and I'm halfway through that movement by the time the pain reaches my brain. Oh, it's a burn. Oh, it's a pain. And that's because of this spinal cord processing in the gray matter. You'll notice here's the receptor of the pain coming into the spinal nerve, splitting to the dorsal root and arriving here in the sensory horn, where we have a synapse that splits the signal. This is divergence. And the longer path crosses over from right to left, ascends the left side, and delivers that pain sensation to the cerebral cortex. But sensation on the right side, pain on the left side. If a motor signal is sent back to the right side, it's going to often come down here, cross over again, get to the ventral root. But the second pathway is the reflex pathway. All it has to do is move a signal right here to this root, the ventral root, and send a direct message out to a muscle bundle. In this case, it's showing my favorite muscle, the biceps brachii, and its principal action, the agonist or prime mover action of the bicep, is to flex the elbow. Now, that occurs much quicker than sending the message up to the brain, getting a cranial reflex to send it back down, and moving the hand. Now, it makes sense, doesn't it? The reflex is not quite involuntary. I know any number of people, and I've seen any number of dramatizations where people would control their reflex action. So the common thing, a person who wants to show that macho quality is they'll put their finger in a candle flame and hold it there while the flesh burns. You can overcome that reflex to draw it back. But under normal circumstances, with normal events, when you put your finger down on a hot surface, you're destroying cells. You normally don't need to think about whether you want to keep doing that or not. And so to have a reflex that jerks it back at the quickest possible speed limits injury. It saves cells, and that's where we find a lot of our reflexes that are hardwired. Reflexes may be innate if we're born with them. They may be acquired if they are learned. Some of our patterns of behavior, like balance, almost becomes an acquired reflex. We don't think about it. We don't direct it. We just, through our internal sensors, we interpret the flow from our peripheral receptors and from gravity in our inner ear, and we basically maintain our balance almost like a subroutine. We can classify them by response. Is it somatic? Is it affecting voluntary skeletal muscles? Is it involving superficial and stretch reflexes? Or is it visceral, autonomic? Are we regulating some smooth or cardiac muscles, glands, or adipose tissue? And that covers a lot of ground. Digestion uses smooth muscle and glandular secretions. The lungs use smooth muscle and also glandular secretions. The urinary tract uses peristalsis, and so on. We can classify reflexes on the complexity of the circuit. Is it one synapse or is it many? Are there hundreds of synapses or even more, a polysynaptic response? The processing site, is it processed in the spine? That is called a spinal reflex, which is much quicker than a cranial reflex, which is processed in the brain. The stretch reflex shown here, the doctor applies every time you go in and have a reflex test sitting on a table. When you hit this tendon, the one right under the kneecap, what it does is it jerks this tendon and this muscle. Now that's going to jerk a spindle receptor. And sensory nerves are wrapped into that spindle. When it jerks, it sends a sensory message and a direct motor response, a reflex message back out to the effector, in this case to the same location of the gross anatomy of the body. In this case, the pointer shows you the side of the quadriceps group. So if you stimulate that quadricep muscle, it contracts and jerks the knee. Now, it's a good test for the continuity of your nervous system and your reflexes in the doctor's office, but what real effect does it have? What real benefit? Well, we're up on two legs. If we collapse one leg, if our knee gives out, or if our foot comes down funny or slips or rolls on a stick, that particular kind of thing can unbalance us to the point where we fall down. Now, if the tripping is not too dramatic, we have the ability to arrest our fall. It's not just to kick. Normally when we're running or walking, we extend our knee as a part of the stride that we're producing. But with the foot on the ground, if we flex the quadricep group, it slows the bending of the knee. Not extending the knee if your body weight's moving you down through a knee that's just kind of gone from straight to bent. But if your knee is collapsing, a reflex stimulation of the quadriceps allows you to, quote, catch yourself on the way down and avoid a fall. This is an enlargement showing you the fibers of the muscle spindle wrapped around this spindle region. This is where the sensory nerves, the sensory region, are affected by a jerk or a shock. Send a message out. If a reflex response is generated, the motor nerves carry it back and basically stimulate these muscles to contract. This is the painful skillet. Now, interestingly enough, this is showing just a light touch, and the biceps brachii is showing this flexor reflex. We're flexing the elbow, but in this case, you notice we did not grasp the skillet handle, because if you reached over to a hot skillet, picked it up, you would have to both flex the elbow and release the skillet. That's a different, more complicated reflex. But this is showing something that has to happen. The flexor reflex is something that actually involves a stimulation of one muscle, in this case the biceps, while at the same time it inhibits the extensor muscle on the opposite side, the tricep. So that allows the elbow to flex with maximum speed if the tricep does not resist it. This gets more and more complicated. This is called the crossed extensor reflex, and I've noticed this before because I used to run quite a bit, and I would step in a hole or put my foot wrong. I would trip on something. And under certain circumstances, the reflex was quick enough. So stepping down on attack, you get that pain reaction, but two things happen at once. You actually are stomping down with your left foot. At the same time, you're drawing up your right foot. And that involves, on the right side, stimulating the flexor of the knee while you inhibit the extensor. On the other foot, you're stimulating the extensor while you inhibit the flexor to produce this almost simultaneously stomp and withdraw, stomp and withdraw. So again, this is related to our bipedal posture and our upright stance on two legs. It's hard to walk on two pegs like our legs. And so the balance and the posture involved, the reflex wiring, is fairly complex. This is a developmental reflex, and it shows a curling of toes in the adult. If a doctor draws a blunt instrument across the sole of the foot in an adult, the normal thing that happens is a curling of the foot around the instrument. So basically what we're doing here is we're pressing down with the toes. We're flexing the arch of the foot downward from the heel position. Now, when we're born and we do that, these are not trained into us, and so the baby's foot does respond, but it responds like this, basically spreads away from the sole of the foot, and that the toes spread out, because they haven't learned about posture and standing, and we don't have that physical program. This kind of test, if you get the dissociative response shown on the right in an adult, it indicates problems in neural connection in the pelvic appendicular skeleton. So to wrap up, I'm going to go back to this kind of central anatomical breakdown that shows you the most about the spinal cord, that shows you the connection of the spinal nerves. Now, out here would be a spinal nerve, this being the left and right, since we're on the posterior side. On each side, it splits into a dorsal root facing you here, and a ventral root. The dorsal root is carrying sensory messages to this span of the dorsal gray horn. Sensory input, the gray matter, or any gray matter, is where you're going to form more synaptic connections and do information processing. And when motor commands are generated that are appropriate to this spinal nerve, they would enter the anterior or ventral nerve elements, congeal on the ventral root, and go out through this spinal nerve. That applies right and left. Now, other things you see are the vertebral case around the complete spinal cord shown at the bottom, but in the middle here, peeling back, the three wrapping layers of the central nervous system: the pia mater, the arachnoid mater, which is actually this little space in here, filled with cerebrospinal fluid, and the tough, flexible, almost indestructible Dura Mater that's on the outside. So this figure is a good one to keep in mind when you're thinking about the organization of the spinal cord. I would reiterate that the spinal cord is organized around the division of the spinal column, the vertebral column, into vertebra. Hard, strong bones that protect the spinal cord, but permit for great flexibility while providing strong and flexible support. So, and